Our laboratory is part of the Wellcome Trust Centre for Anti-Infectives Research, and we study parasites called trypanosomes. These single-cell parasites are transmitted into blood by blood-feeding insects, such as an infected tsetse fly. The parasites cause lethal, neglected tropical diseases in humans and in domestic animals. These and related parasites are generally transmitted in tropical countries in Africa, South America, and Asia. We grow and study African trypanosomes, the parasite, in plastic flasks in the containment lab. We can see them using a simple microscope. Our research looks at how the parasites cause disease in infected people and animals, and how we can find ways to stop this using medicines. A major challenge is that parasites can develop resistance to medicines. This happens when there is a change in the parasite's DNA, their genetic code, called a mutation. One mutation we found stops medicines from entering the parasite. We study resistance to medicines by mutating the parasite's DNA in the lab. We observe and record the effect of the changes we make. We have developed a method that precisely edits the parasite's DNA using small pieces of synthetic DNA. We design the synthetic DNA at the computer, which is then made by a company and delivered to us. Once we have the synthetic DNA, we are ready to do our experiments. First, we need some parasites. These are taken from storage in liquid nitrogen at minus 196 degrees Celsius. They are grown in a nutrient-rich liquid at 37 degrees Celsius to mimic the conditions the parasite would encounter in the blood. This work is carried out in a microbiological safety cabinet in our containment lab to prevent contamination and to protect the scientist. The parasites grow quite quickly, and 1 million thawed cells can produce 100 million cells in just 2 days, enough for several experiments. Once the parasites have grown, we can edit their DNA. We collect the parasites using a centrifuge that separates them from the liquid. We then mix them with the synthetic DNA. To get the synthetic DNA inside the parasites, we zap them with a short, high-voltage pulse of electricity. Afterwards, the parasites are left to recover for 6 hours in a warm, nutrient-rich liquid. During this time, some of the cells will have inserted their synthetic DNA sequence into their genome, causing a mutation. After 6 hours, we add a medicine to kill the parasites. Any parasites that have the synthetic DNA sequence will have become resistant to the medicine. They will survive an increase in number over the next few days. We check any live parasites under the microscope and take a sample of their DNA to check if the synthetic DNA is present. When we have finished our experiment, all waste is treated with a disinfectant and placed in a leak-proof box. These boxes are then heated to 121 degrees Celsius in a machine known as an autoclave, which is like a large pressure cooker. The waste is then safe for disposal. We can study these parasites to help explain how the medicine works and how the parasites become resistant. This new information can help us to design better medicines.